Good morning, students. Today, our topic is gymnosperms. We have completed pteridophytes. This is gymnosperm. So what are these gymnosperms? What is the meaning of gymnosperms? These are under division spermatophyta we know. These are open-seeded plants. These are open-seeded plants. Why those are open-seeded plants? There is no ovary. Where are the ovules? Inside the ovary. From where the seeds come? From the ovules. So inside the ovary, ovules are present. We have seen that in angiosperms. Those ovules after fertilization enter the seeds. But here, there is no ovary at all. Why there is no ovary in gymnosperms? Because the megasporophyle is open. Means the carpel is open. See here, this is a megasporophyle. It is open carpel. This is open carpel. So these are the ovules. These are the ovules. These only will become the seeds. Those are exposed ovules. Now, in the case of angiosperms, this megasporophyle is closed. That's why we can see the ovary like this. Style and stigma. Inside this ovary, we can see the ovules in different types of placentations we have seen. Marginal placentation, axial placentation, parietal placentation, central placentation, like that, the ovules are arranged inside the ovary in the case of angiosperms. But here, megasporophyle is open, it is not closed. That's why there is no ovary at all. Okay, so ovules are exposed. That's why the seeds are also exposed. Naked seeds, open seeds. There is no fruit. Flowers are present, but fruits are absent in gymnosperms. Flowers are present, but fruits are absent. Okay. So what are the living fossils here? Living fossils. Living fossils of gymnosperms. Ginkgo. Biloba. This one. Ginkgo biloba. This is Ginkgo biloba. Then cycas also. Cycas. You see cycas? This one is cycas. This is cycas. This is ginkgo. Both are living fossils only. Next. So what is this sequoia dendron giganteum? That is commonly called as redwood tree of sequoia. Redwood tree of California. The scientific botanical name is Sequoia dendron giganteum. Tallest living plant. Tallest living plant. More than 100 meters height. More than 15 meters girth. And what about the plant body here? In the case of bryophytes, the plant body is gametophyte, we know. In the case of pteridophytes, the plant body is sporophyte, you know. Here also, you can see plant body sporophytic. Plant body is sporophytic. What is the meaning of sporophyte? It consists of root, stem, and leaves. Root, stem, and leaves. Are these tracheophytic? Yes. Like pteridophytes, these are also tracheophyte, means vasculature is present. Embryophytic, like bryophytes, pteridophytes, gymnosperms also, they can produce embryo after fertilization. Means after the zygote develops into embryo. Okay, next. So plant body sporophyte means it consists of root, stem, and leaf. Okay, so what type of root you can see? Taproot system. Like dicots, taproot with many lateral roots. with many lateral roots. So the root of cycas, when you observe, anatomy of the root is similar to that of the dicot root, but in the case of cycas, the general cortex. So what are the parts of the root? Dicot root, epidermis, cortex, and still we know. So in the general cortex, that is also differentiated into outer, middle, and inner cortex. In the middle cortex of the general cortex, you can see the coralloid roots. Look here. These are the coralloid roots. Coralloid roots. Coralloid roots. Coral-like. Coral-like. You see here, this is epidermis and this is the general cortex. So general cortex, outer cortex, middle cortex, and inner cortex. So the middle cortex with the blue-green algae. What are these blue-green algae? Nostoc and Anabina. Nostoc and Anabina. Remember, the coralloid roots of cycas consists of blue-green algae like nostoc and Anabina in the middle cortex or in the general cortex. For what purpose? For nitrogen fixation. Okay. Next. So the root only, this is in a cycas. So cycas root is called as coralloid root because those are coral-like. Cycas, coralloid root. And it consists of nostoc and Anabina. Nostoc and Anabina. Okay. And what about pinus root? We know mycorrhizal roots. They are in association with the roots of higher plants. Means fungal haustoria in association with the roots of higher plants. So pinus roots, the young roots of pinus consist of ectotrophic. You know the mycorrhiza are of ectotrophic or endotrophic or ectendotrophic. We have studied ectotrophic mycorrhizae in pinus, we can say. Okay. So cycas roots are coralloid roots and the pinus roots are having ectotrophic mycorrhizae. Ectotrophic mycorrhizae. And what about the leaves? When you see the stem, cycas stem, you see, it is like coconut, that is cortex. Cortex. No? Like that only you can see the cycas. And the leaves. They show xerophytic adaptations. Leaves show xerophytic adaptations. Important. Leaves show xerophytic adaptations. What are xerophytic adaptations? Xerophytic features. What are those? The most important, thick cuticle. Cuticle is very thick. And stomata. Can you see the stomata? It's on the, means what is the purpose of this stomata? Exchange of the gases means transpiration, we can see. But here they are showing xerophytic adaptations means there is less water. That's why to reduce the loss of water, the cuticle is present and stomata should not be exposed. That's why those are in the sunken pits. So sunken stomata. So because of this thick cuticle and sunken stomata, reduce the water loss through transpiration. Along with these two characters, two features of xerophytes, they have some other features also. Parenchyma is not, normally parenchyma is like this, no? But here the parenchyma is armed parenchyma. We can see the armed parenchyma. And transfusion cells are present. And the albuminous cells are present. These are all xerophytic adaptations in that's in gymnosperms. The leaves, they are showing xerophytic features. Okay. So all these characters, they help to withstand extreme temperature, humidity, and wind. extreme temperature, humidity, and wind. And what about the these vascular tissues? Xylem. We have studied in pteridophytes. Xylem consists of pteridophyte. Xylem has only tracheids. Here also you can see only tracheids. Only tracheids. But we can see vessels in Neetum. Neetum is also one of the example of gymnosperms. Here we are studying about the gymnosperms. Gymnosperms example Cycas, Pinus, and Neetum. Neetum is advanced type. Advanced. So there you can see the vessels also. Okay, then phloem. What we have seen in the pteridophytes in the phloem, sieve cells. Here also you can see the sieve cells, not the sieve tubes. And companion cells are absent there. Here also companion cells are absent. There means what? Pteridophytes. Here also companion cells are absent. Albuminous cells are present. Albuminous cells are present. Okay. Then come to the reproduction. Reproduction. In bryophytes, what about the spores? Homosporous or heterosporous? Homosporous. In pteridophytes, some are having homosporous, majority, and some are having these heterosporous condition. Okay. What are the examples for pteridophytes having different kinds of spores? Selaginella and salvinia. Here, all are heterosporous. Heterosporous. Two kinds of spores we can see. What are these two kinds of spores? Microspores. From where they are produced? From the microsporangia. Where are these microsporangia? On the microsporophylls. On the microsporophylls. So microspores are nothing but pollen grains only. Those are developed inside the microsporangia. Microsporangia are present on the microsporophylls. And megaspores, another type of spores, megaspores. Where they are produced? Inside the megasporangium. That is the ovule. Integumented megasporangium is the ovule. There we can see the megaspores. These megasporangia are present on the megasporophylls. Megasporophylls. What are these sporophylls? We know pteridophytes. Sporangia, spore-bearing leaves are called as sporophylls. If they bear microspores, microsporophylls. If they bear megaspores, megasporophylls. So here these sporophylls aggregated to form. Sporophylls aggregated means grouped to form strobili or cones like that of equisitum and selaginella of pteridophytes. Strobili or strobilus singular. They may be lax or compact. Lax means some loosely arranged or compactly arranged. Okay. So flowers are naked except in a Neetum. Flowers are naked. What is the meaning of naked flowers? Flowers are naked. Seeds are naked, we have seen now. Because megasporophyll is open, that's why ovules are exposed. There is no ovary at all. There is no fruit at all. That's why the seeds are naked seeds, open seeds. Flowers are naked means no perianth parts. But in Neetum, you can see perianth parts. So, flowers are naked except in eclamadious flowers. We use the word eclamadious. In Ceratum inflorescence, we have studied eclamadious flowers. Eclamadious. A means absent. Perianth parts are absent except in Neetaceae or Neetum. Okay. So, the strobili bearing microsporophylls Microsporophylls and microsporangia are called microsporangiate. Sprawly bearing megasporangia, megasporophylls called as megasporangiate. When you see the pinus, you see this stem, cycas stem, I said codex means unbranched. But in pinus, the stem is branched. Pinus as well as in seedless, the stem is branched. You see the diagram of pinus. Look here, this is pinus. See, stem is branched. The same as citrus also. In the case of cycas, the stem is unbranched. Codex, coconut, like a coconut stem, codex stem, unbranched. But in pinus, you can see branched stem, and citrus also branched stem. Okay, so here, monoecious. Pinus is monoecious, cycas is dioecious. Pinus is monoecious, cycas is dioecious. What is the meaning of monoecious? So Pinus is the example. Means male and female sex organs are on the same plant. That is example Pinus. And what about dioecious? Male and female means microsporophylls and megasporophylls are on different plants. Male plant and female plant. Microsporophylls on male plant, megasporophylls on female plant. Okay, so this is cycas example. In Pinus, microsporophylls, they form male cone. Megasporophylls, they form female cone. Okay, both are present on the same plant. That's why this is monoecious. When you observe cycas, remember, when you observe cycas, there is male cone. Microsporophylls, they form male cone in the male plant, but female cone is absent. Very important. Female cone is absent in cycas. But in Pinus, you can see both the male cone and female cone on the same plant, monoecious. Whereas in cycas, that is dioecious, male and female plants are different. Means male and microsporophylls on one plant, megasporophylls on female plant. So microsporophylls, they form the male cone. Look here, this is the male cone of cycas. Cycas male cone. See, all the microsporophylls, they are aggregated to form a cone-like structure. This is the cone-like structure. Okay, that's why those are called as cones or strobili. But again, in the case of the cycas, a female plant, there is no female cone. Why? All those are arranged loosely. See, these megasporophylls are arranged loosely. That's why there is no formation of female cone. Remember, in cycas, there is no female cone. Cycas is dioecious. Cycas is dioecious. Now, you see the microsporophylls, how they form the male cone. Okay, this is the male cone. When you see all this, these are all microsporangia. This is one microsporangium. Okay, so many microsporangia. See, when you cut this, you can see like this. Many microsporangia. So see, how is this male cone is formed? By the compaction of, means the close association of all the microsporophylls. Okay, and you see the this is the microsporophyll. This is the male cone. Okay, and these round structures are microsporangia. Look here, so many microsporangia are present. Where do you see many microsporangia on the undersurface of each microsporophyll? This is cycas microsporophyll we are seeing. Undersurface of the microsporophyll, we can see many sporangia. And what about in Pinus? Here you are seeing many microsporangia. Look, so many are there. But in the case of Pinus, only two means conifers. Coniferaceae, Pinus under the class Coniferopsida. Okay. So this Pinus consists of, or conifers consists of, or Pinaceae consists of only two sporangia. Only two sporangia. Look here. This is a microsporophyll. Okay. These are the microsporangia. These only two microsporangia. These two are the two microsporangia. But here so many microsporangia present on the microsporophyll in the case of Cycas, but in Pinus only two sporangia or microsporangia. Okay. So inside the sporangia, what is present? We know the spore mother cells, microspore mother cells, microspore mother cells. These microspore mother cells, they undergo meiosis. What they form? Microspores. Microspores. Okay. So what are these microspores? They develop into male gametophyte. What is that male gametophyte? Nothing but pollen grain. Male gametophyte. Okay. In the case of gymnosperms, male gametophyte and female gametophyte are highly reduced. So this highly reduced male gametophyte consists of only one protalial cell in the case of Cycas and two in the case of Pines. We see in our NCERT they haven't mentioned is the structure of this male gametophyte. Okay, but male gametophyte is highly reduced. All these points are very, very, very important. Okay, so microsporophylls are undergoing meiosis and they form the microspores. Microspore mother cells are diploid. Okay, that is sporophytic generation only. They undergo meiosis and they form haploid microspores. It develops into male gametophyte, that is pollen grain, which is highly reduced and parasitic on the sporophyte. Highly reduced and parasitic on the sporophyte. So now when you see the male gametes which are produced by the male gametophyte, are they motile? In the case of male gametes, in the case of Cycas, those are motile. That's why pseudogamous type of oogamy you can see in the case of Cycas. But in the case of ginkgo, sorry, Cycas as well as ginkgo also. And Pinus and NEET, those are non-motile. Cycas and Neetum are living fossils, I said, no? So the male gametes of Cycas and ginkgo are motile. That's why you can see pseudogamous type of oogamy. What is pseudogamous? Fertilization after the fusion of the male and female gametes, we know. The male gametes, if they are motile, with the help of the motile organs, if they move towards the female sex organ and when it fertilizes, we call that as pseudogamous type of oogamy. Okay. So in case of Pinus as well as Neetum, we can see non-motile gametes. That's a what type? And the pollen grain, it forms the pollen tube. Means the pollen tube is carrying the male gametes, like angiosperms. So this is siphonogamous type of oogamy. Siphonogamous type of oogamy, where the gametes, when they are non-motile, means Pinus and Neetum, siphonogamous type of oogamy. Whereas in Cycas and ginkgo, pseudogamous type. Already the meaning I said in pteridophytes as well as in bryophytes, pseudogamous type of oogamy in Cycas and ginkgo. And in Pinus and Neetum, siphonogamous. What is siphon tube? The tube is produced by the pollen grain after germination, and it is carrying the male gametes which are non-motile. Okay, so this is about the male microsporophylls, male cone, and microsporangia, male gametophyte, male gametes. Okay, then come to female cone or female flower. How is this formed? With the help of the megasporophylls. Female cone or female flower. This is only called as flower, but here there is no flowers are present, but no fruit. Female flower. What it has? Megasporophylls. Megasporophylls. Can you see the female cone in cycas? No, already I said. Female cone absent in cycas, but you can see in a pinus. Okay. So, megasporophyll. Normally, what are the other names for megasporophyll? We can call this as carpel. We can call this as gynoecium. We can call this as pistil. This is 4 of the synonyms, means megasporophyll, pistil, gynoecium, carpel, all are same only. We know very well in angiosperms. Angiosperms. So, the gynoecium consists of ovary, style, and stigma. Inside the ovary, what are present? Ovules are present. Here, the megasporophyll already I showed like this. This is the megasporophyll. Okay, here you can see the ovules. Can see the ovules. So, what about the type of ovule in the case of cycas? Here, largest ovule we can see. Largest ovule. Where? In cycas. And in the cycas, you can see orthotropous type of ovule. Orthotropous ovule. And in case of pinus, anatropous ovule, we can see. Cycas, orthotropous. What is the meaning of orthotropous? What is the position of micropyle, chalaza, and funicle? All are in a same straight line. Orthotropous ovule in the case of cycas, and in pinus, anatropous ovule. What is anatropous? Inverted ovule. Anatropous ovule in case of pinus. Okay. So, integuments. So, you know the structure of the ovule. This is the nucellus. Okay. Nucellus consists of, see, what is the ovule ploidy? It is also a sporophytic generation only. No? Diploid. So, these are all nucellar cells. This is nucellus, diploid. It is diploid. All the cells are diploid. Surrounding this, what is present? Integuments are present. When you see in NEET, it is a bitegmic. Normally unitegmic. In cycas and pinus, only single integument is present. Whereas in NEETM, you can see 2 integuments, like that of angiosperms. Okay, NEETM is advanced type. So you can see the integument, unitegmic in cycas and pinus. Okay, surrounding the megasporangium. Now we call this as what? Ovule. Okay. This is nucellus, and this is the micropyle. This is the chalaza, and this is the funiculus or funicle. Okay, all are on the same straight line. That's why this is called as orthotropous ovule. Okay, now listen carefully. So this nucellus is diploid. Nucellus is diploid. In during fertilization of angiosperms, we have studied the development of the female gametophyte or embryo sac. Okay. Here how the female gametophyte is formed. Here we don't call female gametophyte as embryo sac. We call here the female gametophyte as endosperm. Remember. So one of the nucellar cell. Look here. One of the nucellar cell. Act as megaspore mother cell. Megaspore mother cell. So many cells are there now. One is acting as megaspore mother cell. Diploid. Okay. So megaspore mother cell is undergoing meiosis. And it forms megaspores. How many? 4. Same like that of angiosperms only. Okay. It forms 4 megaspores. These are the megaspores. 4 megaspores. Okay, 3 degenerated, only 1 is functional. See, this may be monosporic or tetrasporic also. Okay, we are seeing the monosporic type C. So, 1 is functional like that of polygonum of our angiosperms. So, one of the megaspore that is functional, chalazal megaspore is functional. What is happening in the megaspore, that functional megaspore? So, this is the megaspore. Bigger one I am showing. This one. All these 3 they degenerate. Only 1 is functional. Only 1 is functional. This one only functional. This only I am showing. So this is the megaspore. So in this megaspore haploid, this megaspore is haploid. Okay. This megaspore is nucleus is undergoing 3 nuclear divisions. This undergoes first mitosis. That is division only. Nucleus. And these also undergo again. So each one is undergoing 3 nuclear divisions. Okay. So multinucleated structure is formed. Now wall formation takes place. Surrounding each nucleus, wall is formed. Now this is called as the endosperm. This is called as the endosperm. Is there any fertilization now? No. So this is a pre-fertilized tissue and it is formed from the megaspore. Okay. 3 nuclear divisions taken place inside the megaspore. Cell wall formation takes place. It will become the endosperm. Okay. It is developing from the haploid megaspore. That's why it is a haploid. It is a pre-fertilization tissue. This endosperm is called as female gametophyte. Female gametophyte. Remember, this is called female gametophyte. In gymnosperms, endosperm is called as female gametophyte. So this normally female gametophyte has female sex organs. What are those female sex organs? Archegonia. How many archegonia are present? 2 or more archegonia are present in the female gametophyte. What is the archegonium structure? I said already, gradual reduction. The archegonium you can see from bryophytes to gymnosperms or angiosperms. See, in the bryophytes, the archegonium is flask shaped with a stalk, venter and neck, I said. And in the case of pteridophytes, venter and neck only present. And in the case of gymnosperms, only venter is present. Only venter is present. And in the case of neetum also, neetum is advanced type, no, archegonium is absent. And in the case of angiosperms, no archegonium at all. So only venter is present. How many are there? 2 or more archegonia. 2 to 8 you can. Okay. So inside the venter, what are present? Female gamete is present and ventral canal cell is present. This is the mouth of the venter. Okay, so like this, archegonia are present with the egg and ventral canal cell. So here inside the ovule it is all taking place. Remember. So here this is the endosperm. This is the endosperm. Haploid female gametophyte with archegonia. Archegonia having only venter. Venter is having egg and ventral canal cell. Okay, so this is the archegonial chamber. Now, how is the fertilization taking place? We see. Now, come to the pollination. See here, one more point. Male gametophyte and female gametophyte in gymnosperms are not independent. Where are those present? In the on the sporophyte? Still means lifelong they are present on the sporophyte only. See, female gametophyte, male gametophyte, those are not independent. But in bryophytes, plant body itself is the gametophyte. It is showing independent life. And in pteridophytes, the prothallus, that is small but green. It is free living, independent. But here in gymnosperms, these are not these are not independent. Those are dependent on the sporophyte, remember, retained in the sporangia of sporophytes. This is the megasporangium. Ovule is the megasporangium, no? Integumented megasporangium. Inside that only female gametes you are seeing. Okay. So this point is very important. Male gametophyte and female gametophytes are not independent in gymnosperms. But in pteridophytes and bryophytes, you can see independent gametophytes. Okay. Here pollination is direct. What is indirect pollination? We have seen that in angiosperms. Ovary, style and stigma. Ovary, style and stigma. Where? In angiosperms. Ovary, style and stigma. Okay. The pollen grain, through different methods of pollination, they fall on the stigma like here. Okay. In 2-cell stage, majority 60% 2-cell stage and other 3-cell stage we have studied in angiosperms. Okay. So it is falling on the stigma, then the pollen tube germinates and it passes through the style and reaches the ovary. Then inside the ovary, ovules are present, then it enters into the ovule. Now, this is indirect pollination. But in the case of gymnosperms, there is no ovary at all. There is no ovary. No, that's why directly they fall on the micropyle of the ovule. This is the micropyle of the ovule. How they come? What is the type of pollination? This is called as direct pollination. And what is the type of pollination here? What is the agent here? The wind is the agent. Through air currents, these pollen grains are carried and they fall on the micropyle of the ovule. Micropyle of the ovule. So opening of the at the mouth of the ovule. Mouth of the ovule means micropyle of the ovule. Then the pollen grain germinates. Here the pollen grain germinates and it forms the pollen tube. This pollen tube enter into the, so the pollen tube is carrying the male gametes, we know. So the pollen tube which is carrying the male gametes, it reaches the archegonial chamber here. This is the archegonial chamber. Those are waiting, the female gametes are waiting for the male gametes. Okay. So now the male gametes are released. Those are sucked by the mouth of the venter. Okay. And they enter into the venter, fuses with the egg, forming the zygote. This is very important. This question also came So these pollen grains are carried in air currents and come in contact with the opening of the ovules. Ovules born on the megasporophylls. The ovules are born on the megasporophylls. The pollen tube carrying the male gametes grows towards the archegonia in the ovules. Where are the ovules? Where are the archegonia? In the ovules, and discharge their contents near the mouth of the archegonia. Near the mouth of the archegonia. See, the pollen grains are carried by air currents. They fall on the micropyle of the ovule. Their germination takes place and they form the pollen tube. It grows towards the archegonia, the female sex organs, and they discharge their content into the archegonia. Archegonium is having only venter. Okay, like this, the fertilization is taking place. So what is formed? Zygote is formed. After fusion, zygote is formed. The zygote develops into, zygote is diploid. Zygote develops into embryo. Okay, you can see in Pinus polyembryon, you can see. So embryo has how many cotyledons? In cycles, 2 cotyledons. But in a Pinus, many cotyledons. Okay, seeds, they may undergo dormancy. Okay, here what is dominant here? Sporophytic generation is dominant. Sporophytic generation is dominant, and gametophytic generation is reduced. Gametophytic generation is reduced. What type of life cycle you can see? Diplontic life cycle. Seeded plants, you can see diplontic life cycle. Okay, so this is about the gymnosperms. Okay, thank you.